Hello, welcome to Chapter 9, Outside Diagnostic Laboratory Services. In this chapter, we will identify the correct sample needed for specific testing, list methods used to preserve and label samples correctly. We will go over laboratory forms and how to submit samples safely to the lab. Finally, we will cover how to create an appropriate fee structure for outside laboratory services. Many practices continue to use in-house laboratory services for general chemistries and complete blood counts, or CBCs. For all tests that cannot be completed in-house, an outside service must be chosen. The diagnostic laboratory must offer services and tests that the veterinarian is looking for. Services include analysis for biochemical profiles, CBCs, cultures, cytology, and histopathology. Laboratories should conform to guidelines set forth by the National Committee for Clinical Laboratory Standards and the FDA. Patient samples must be prepared according to lab standards and sent correctly to the lab in order for accurate results. You must understand the difference between serum and plasma when following the sample submission requirements for laboratory tests. Serum is the liquid portion of the blood that remains after the blood has clotted. Serum is produced when red top tubes are centrifuged, separating the red blood cells and coagulation proteins from the liquid portion of the blood. There are two ways to collect serum. A technician can draw a sample and place it into a red top tube. Let that sample clot naturally for 15 to 20 minutes. Spin it in a centrifuge and then pipette the serum off the top to put into a plain red topped tube for transport. Another option is to use a serum separator tube, which has a plug that separates the serum during the centrifuge process. Although the second option seems easiest, there are certain tests that you cannot use a serum separator tube for. Plasma is the liquid portion of the blood that contains clotting factors. There are several anticoagulants used in blood tubes to keep it from clotting, thus preserving the plasma. Plasma is obtained by centrifuging the sample and removing the liquid portion of the sample without any red blood cells. EDTA is used in lavender-topped tubes. Lithium heparin or sodium heparin are used in green top tubes. Potassium citrate is found in blue top tubes. When completing a laboratory blood submission, you must pay careful attention to the tubes you need to fill. As you can see from this chart, many tests require serum, but some specifically state to use a red top tube. Others require a tube with a specific anticoagulant like EDTA or potassium citrate. Knowing how to store the collected sample is just as important. Most samples require refrigeration, although a blue top tube requires freezing. Each sample, including blood tubes, biopsy jars, and slide containers, must be clearly marked with the patient's name, date, and the type of serum, like the type of specimen, like urine, serum, plasma. All lids and all caps should be secured to prevent the sample from leaking or formalin from spilling. A protective, absorbent material should be wrapped around individual samples to prevent the sample from being broken during shipment. Slides should be packaged in a slide container to prevent the glass from breaking. Formalin is a preservative that maintains the tissue characteristics for transport. Larger tissues may be partially cut, allowing the formalin to penetrate thicker tissues. The specimen container should contain 10% formalin at 10 times the volume of the tissue. If a sample does not have enough formalin, then it will not be fixed or preserved for histopathy. Do not reuse jars because they may contain residual particles of the previous specimen and are often labeled with the old patient's information. Formalin should be used with caution because it is a known carcinogen. It should not be inhaled, and gloves and eye protection should be worn when handling the chemical. Most laboratories provide pre-filled formalin jars. If formalin is supplied in a gallon container, jars should be filled under a fume hood or in a well-ventilated area. Laboratory procedure books should be consulted before sample preparation to ensure the correct cytology is submitted. Cytology samples may require staining depending on the test or the facility. Cytology slides should never be shipped in the same bag as formalin containers because the fumes are known to degrade cytology samples. The veterinarian should indicate which culture is preferred depending on the location of the sample. Samples submitted for cultures are either anaerobic or aerobic. Anaerobic is a technical word that literally means without air, whereas aerobic refers to with air. Aerobic samples should be kept refrigerated until pickup and then shipped with a cold pack. Anaerobic cultures should be kept at room temperature and processed within 48 hours of sample collection. As with any sample, be sure to label them with the patient's name and the date. Figures 9.2, 9.3, and 9.4 in your textbook show a few examples of laboratory forms. Be sure that you fill them out with all of the pertinent information regarding the patient's signalment as well as complete medical history. Some forms are computerized and integrated into a practice's medical record software. Practices with electronic medical records may choose to have results automatically populated into the medical record. Alerts are set for the team when results are available. Other specialized labs may require you to use a paper form. Copies of submission forms should be kept for record keeping and tracking results. Samples may become lost in the mail, the wrong test may be performed, or results may not have been received. Proper sample shipment is absolutely critical when submitting samples. Keeping the samples chilled preserves cell function. This is especially important during Arizona's hot summer months. Samples may be submitted with a bag of hot saline solution and can be insulated by wrapping them in layers of newspaper or bubble wrap to prohibit freezing. Team members should verify shipping procedures with the laboratory to ensure the best sample submission and results turnaround time. Some shipments may need to be sent overnight to the lab. Some labs nearby will send a courier to pick up the samples. It is ideal to have samples picked up late in the day, allowing any samples collecting during the day to be sent. Ideally, results should be received the following morning. Patient results may be reported by fax, phone, or internet. Reference ranges indicate the normal ranges for that particular piece of equipment. Flags can also indicate patient results outside the normal range, drawing attention to specific abnormalities. Studies indicate that red blood cell morphology and platelet parameters change with delayed analysis. If a patient's CBC appears abnormal, a pathologist should review a blood smear. Laboratory reports should be detailed and provide current information for the veterinarian. This service saves the veterinary team time when lab reports include specific disease and treatment indicators with regard to sample results. Reviewing test forms for turnaround time helps prevent a doctor or client from becoming upset. Specialized tests may take longer to run. However, if results are delayed, a lab representative should call the practice and communicate the delay. For the practice to recover hidden costs associated with laboratory analysis, the practice must at least double the cost of the testing to the client. A team member can call the lab and receive specific pricing information as well as details on shipping the sample and the turnaround time for results. This can save the practice and client money, along with being able to receive results quicker. Appropriate fees must be collected from clients for laboratory services and the cost of supplies, shipping, and interpretation of results. Team members often place calls looking for specific results, lab codes, or supplies. It is important that the call is answered in a timely manner, that lab personnel are friendly, and that they provide the answers needed. Veterinarians often need to consult with a pathologist or specialist regarding cases and results of previous submitted blood work or lab work. Most laboratories will provide veterinary practices with supplies for submitting samples free of charge. Supplies may include blood tubes, histopathology jars, sample submission bags, and forms. If any additional materials are needed, a call to a customer service representative should take care of the request. This concludes the lecture for Chapter 9.